Greetings to all registered students from Gujarat State Biotechnology Mission. Myself, Dr. Actin Mukherjee, Assistant Professor and Head of the Department, Shri P.M. Patel Institute of Postgraduate Studies and Research in Science, affiliated to Sardar Patel University, Anand. The title of my today's invited talk is Cytoskeleton. Under the aegis of Gujarat State Biotechnology Mission, the session conducted by Navratna University, Vadodara, as a part of GSBTM's initiative, rather I would say a novel initiative of weekend e-lecture series. So dear students, first I shall brief you about the chapter outline. This chapter, in this video, I shall brief you about three important aspects. Microfilaments, also called as actin filaments, shall feature the first segment of my talk. This in fact is more than 50% of the allotted time. Thereafter, I shall focus on microtubules, and towards the end, I shall close my talk with intermediate filaments. So this is the exact flow that I would like to go. At the very outset, let me give you an overview of major functions of the cytoskeleton. When we talk about the major functions of cytoskeleton, 5 important functions come to our mind. The first and the foremost, structure and support. The structure and support to the cell is provided by cytoskeleton. It helps in intracellular transport, trafficking of material from one part of the cell to another. The movement of the cell, also called as motility, and contractility is also one of the function of cytoskeleton. Cytoskeleton is a spatial organizer. Besides, cytoskeleton also has a very important role as far as the cell division is concerned. This summarizes the overview of the major functions of the cytoskeleton. In the next segment, I shall move to the structure followed by organization of microfilaments, and I would stick to actin as the representative example. So structure and organization of actin filaments, and my example for today is structure of microfilament, the actin filament. If you see the diagram, just like Watson and Crick's double helical DNA, here also you can find that there are twisted or intertwined double stranded structure. The yellow highlight is one and the green highlight is another. So there are two twisted structures. These twisted structures of microfilament, especially actin, they play a very important role in muscle contraction, and besides muscle contraction, it also gives structural support to the cell. I would like to brief you that whatever we are studying currently in terms of microfilament, that is actin, most of the research took place from muscles way back in 1940s. I would also like to place on record that My alma mater, that is the Maharaja Sayaji Rao University of Vadodara's Zoology Department, was also known for muscle physiology. The first head of the department, Professor J.C. George, was a postdoctoral student of Albert St. George, the Nobel laureate, as well as Sir Hans Krebs, and one of the thrust area of the department was muscle physiology. Why I said this is because actin that we are studying today in 2021, the actin was also isolated from muscle cell way back in 1942. As at the present perspective, we know that in muscle 20% of the cell protein is actin. But then the concept of actin has changed over the years. So I will talk about actin then and actin now. One important point is that actin is uniquely involved in muscle contraction. So what's the latest from actin? The latest from actin after years of research is that it is an extremely abundant protein. Approximately 5 to 10% of the total protein is actin. Even the prokaryotes such as yeast also do have actin. In fact, yeasts have a single actin gene. Talking about mammals, there are 6 distinct actin genes, of which 4 are muscle specific and 2 are non-muscle specific. Even the cytoskeleton has a strong evolutionary perspective. There is a evolutionary aspect to cytoskeleton. In fact, when you talk about evolutionary perspective, we take account of yeast actin. In case of yeast, 90% similarity exists in terms of the amino acid sequence to mammalian actin. We also have MREB protein. In fact, this MREB protein is a prokaryotic ancestor of actin, and again this is responsible for the structure of several rod-shaped bacteria. What I essentially want to emphasize from this slide, that don't be under the impression that cytoskeleton, especially actin, is just limited to the higher organisms. If you go deep down the phylogenetic lineage, even you have MREB protein in prokaryotic ancestors of actin. So this gives an overview of evolutionary perspective. Now when we come to the structure of actin filament, actin is a monomeric unit forming of actin is G-actin. It shall be better if I focus on the diagram. The monomeric form of actin is also called as the G-actin. Then you have a linear chain of G-actin subunits. Many G-actins will join linearly to form a filament structure that I have highlighted just now. This filamented structure, because of the filament, is called as F-actin. So G-actin joins together to form F-actin, and actin molecule also contain magnesium ions, and this magnesium ion is complexed with either adenosine triphosphate, that is ATP, or with adenosine diphosphate, that's ADP. Since you have G-actin, the monomeric form, and the F-actin, the dimeric form, total you can have the following kinds of actin. Essentially, in terms of ADP, you have ADP G-actin and ATP G-actin, that is two forms for G-actin, ATP and ADP. Likewise, you also have ADP F-actin and ATP F-actin. So, total 2 plus 2, you have 4 types of actin, out of which the ATP G-actin highlighted in yellow and ADP F-actin, these two are the predominant actins in the cell. So, in examination or in entrance exam, if you have a question that which are the two variants of actin which are predominant or maximally present in the cell, your answer would be ATP G-actin and ADP F-actin out of total 4 types of actin that is present in the cell. Once again, to recap, the actin monomers, that is G-actin, polymerize to form actin filaments, that's F-actin. The first step is the formation of dimers. Dimers in turn grow or join end to end by addition of monomers to both ends, and the dimers would form trimers. Trimers in turn would give way to the filamentous structure of F-actin. Again, the structure of actin has 2 ends. You have a minus end and a plus end. So, you have minus and plus end. Later, of course, we will give one more notation to this plus and minus ends as barbed ends and pointed ends. Now, actin is a double stranded helix. The question arises in our mind, why actin filaments have characteristic appearance of double stranded helix? Why actin filaments have characteristic appearance of double stranded helix? The answer lies in the way the monomers are rotated across the scaffold. Since each monomer of actin is rotated 166 degree in the filaments, we have the actin which is a double stranded helix. With this explanation of the G-actin, which forms dimer followed by trimer and followed by the filamentous appearance, it's time for us to focus on the polarity of actin filaments. The polarity of actin filaments, at the very outset, the actin filaments are oriented in the same direction. They are oriented in the same direction. The actin filaments have distinct polarity. As I said, the plus end, also called as the barbed end, and the minus end, also called as the pointed end, are distinguishable from one. It's interesting to focus on how actin behaves in solution under in vitro condition. Under in vitro condition, actin filaments will polymerize spontaneously. So, what you do, first you take a low ionic strength solution. In low ionic strength solution, the actin filaments will depolymerize to monomers. You repeat the experiment, but this time you take a increased or high ionic strength solution, that is to a physiological level. If you take a high ionic strength solution, the actin filaments will polymerize spontaneously. This phenomenon of polymerization and depolymerization also leads to what we will be studying in a short while from now as treadmilling. But before we study treadmilling, let's understand what exactly is actin nucleation. Students, the first step in actin polymerization is known as nucleation. Nucleation is a step which sees the formation of actin nucleus. Essentially, the nucleation involves complex of actin monomers to actin dimer. Actin dimer ultimately post nucleation will form the filaments which will elongate. So, actin nucleus is essentially a complex of 3 actin monomers, and these 3 actin monomers will form a actin filament, and after formation of actin filament it would continue to elongate. As far as the growth of actin filaments is concerned, the reversible addition of monomers does take place at both ends. However, the rate is different. When it comes to the barbed end, also called as the plus end, the elongation in the barbed end is much greater. In fact, elongates 5 to 10 times faster than the pointed end. So, first we have to remember the concept that barbed end or the plus end elongates 5 to 10 times faster than the pointed end. We also have a role of ATP in this perspective. So, what's the role of ATP? The actin monomers bind to ATP. Later, the ATP is hydrolyzed to ADP. That is when the filament assembly takes place. So, what would result? You would result into, suppose this is the entire filament, you have the plus end or the barbed end followed by the pointed end or the minus end. As we see later, you have the barbed end or plus end where the monomers are bound to ATP. As you move from plus end to minus end or from barbed end to pointed end, barbed and this is P pointed, you have the ATP hydrolysis taking place. So, in the initial part it will be ATP followed by ADP bound actin in the subsequent part. So, ATP is not required for polymerization. So, actin monomers that are bound to ATP will polymerize more readily than the actin monomers that are bound with ADP. So, when it comes to polymerization, ATP bound monomers are better favored than ADP bound monomers. But when it comes to depolymerization, the ADP bound monomers are more susceptible to polymerization as compared to the ATP bound monomers. So, this has to be kept in mind. The process of addition of actin filaments is a reversible process. When I say it's a reversible polymerization, that actin polymerization is a reversible process. Why it's a reversible process? At both ends, that is at the barbed end as well as the pointed end, or at the plus end or the minus end, the monomers can be both added, that is polymerization, as well as removed, that is depolymerization. So monomers will associate and dissociate from both ends of the actin filaments. Please remember, it's not subject to only a particular end. At association and dissociation can happen from both ends of the actin filaments. However, the rate of subunit dissociation is independent of monomer concentration. The rate of subunit dissociation is abbreviated as K off, which is independent of the monomer concentration. Now, when we talk about reversible polymerization of actin filaments, the rate of subunit association is proportional to the concentration of free monomers, and this is given by mathematical formula C multiplied by K on, where C is the concentration of free monomer. Next, we have the concept of treadmilling, but to better understand the concept of treadmilling, we will first focus on animation from the cell a molecular approach by Cooper from Oxford University Press. One of the reason this particular animation is free available from the website of Cooper, and I shall depict a small portion right in front of you with our projection. This will help you to understand treadmilling in a better or focused manner. Let's understand the concept of actin monomer polymerization and treadmilling by looking at the animation which I have taken from the Cell, a Molecular Approach, 8th edition, from chapter 14, especially for the figure 14.4. I will just provide you the web link for this animation. Remember students, once again, while projecting this video to you, I acknowledge Cooper's 8th edition of Cell Biology for the content. This content is available free from website under the heading of Student Resources. Let's look at the animation to understand the concept in much better way. The major cytoskeletal protein of most cells is actin. Individual actin molecules, also called G-actin, are globular proteins of 375 amino acids. G-actin is described as having a pointed end G-actin is described as having a pointed end, that is minus end, and a barbed end, also called as a plus end. G-actin monomers polymerize to form actin filaments. The assembly begins with the nucleation of 3 monomers to form a trimer. The polymerization is reversible and proceeds from both ends, yielding a filament called F-actin. Polymerization is a reversible process in which both monomers or both associate with and dissociate from both ends of the filament. At high concentrations of free subunits, the filament grows at each end, but the growth is faster at the barbed end. When the concentration of free actin monomers falls below a certain level, the barb will continue to increase in length, but the pointed end will decrease in length. This results in a phenomenon called as treadmilling. Treadmilling can be depicted in a cycle diagram in which there is a net gain of subunits at the barbed end and an equivalent net loss of subunits on the pointed end. The treadmilling cycle depends on ATP, which is bound to monomers that add to the ends of the filament. Actin monomers at the barbed end are therefore bound to ATP. However, soon after polymerization, ATP is hydrolyzed to ADP. Look, it gets hydrolyzed to ADP, resulting in a zone of ADP bound monomers towards the pointed end of the filament. Remember, ADP bound actin, which is primarily at the pointed end of the filament, dissociates more readily from the filament than ATP bound actin. In contrast, actin bound to ATP associates more readily with the rapidly growing barbed ends. Thus, ATP binding and hydrolysis play a key role in the dynamic behavior of actin. Having seen the animation, now it's time to come back to this treadmilling diagram. So let's recapitulate the major points from this diagram. That this is the plus end, or also called as the barbed end. This is the minus end, also called as the pointed end. If you see this plus end, you have the concentration of free actin monomers. The actin monomers at the barbed end are bound to ATP. However, soon after polymerization, look, ATP, so many ATP bound dimers, they are being polymerized. However, soon after polymerization, ATP is hydrolyzed. Look, loss of phosphate, ATP is hydrolyzed, that is hydrolysis of ATP. Once ATP is hydrolyzed, it results in a zone of ADP bound monomers. There it is, ADP bound monomers. It results into a zone of ADP bound monomers. And where it forms? This forms towards the minus end or the pointed end. ADP bound actin, which is primarily at the pointed end of the filament, dissociates more readily than the filament that is ATP bound actin. In contrast, actin bound to ATP associates more readily with the rapidly growing barbed ends. That summarizes the concept of treadmilling in actin filaments. Dear students, now it's time to understand the dynamics of actin nucleation after clearing our concept from the animation. What exactly we are doing? We are studying the entire process graphically. Initially, at time 0, we are adding actin subunits. Literally, this process wherein the actin subunits, as we saw from the animation, will initially form a dimer followed by a trimer. In other words, this entire process is called as the nucleation. So you have a small nucleus that is formed. It's more than trimer, so we name it as oligomers. Remember students, this entire phase, that is from a single actin monomeric unit to the formation of multiple oligomers or nucleus, this is a time investing process, so it's also called as lag phase, or nucleation phase is also called as the lag phase. Now in lag phase, we already have the nucleus that is formed. Remember students, in this graph on x-axis or abscissa, we have time after salt addition. On y-axis or ordinate, we have percentage of actin subunits in filaments. After nucleation, we move to the elongation phase. Multiple nucleus will join. They will keep on joining to attain the filament formation. So once the filaments are formed, multiple nucleus will join together. So we have growing of the actin filament. That phase where the continuous growth of actin filament takes place is also called as the elongation phase, the second phase in this graph. Elongation phase continues, and finally we come to this state wherein the actin filaments within subunit are continuously being added as well as being removed. This is also called as a steady state where actin filaments with subunits are coming on and off. So this is the final stage of this nucleation graph. Let's do another experiment. In the next experiment, what we will do, we will remove this initial lag phase. So what we are doing, we don't allow the actin subunits to naturally form the nucleus. Rather, what we are doing is, in this diagram, we are already adding preformed filament seeds. That is, the filaments are added from the previous experiment directly. What's happening? The initial lag phase is not there. So we directly start from elongation phase. So we have the elongation phase, the growth of actin filaments, followed by the actin filament with subunits coming on and off, that is the steady state. Having studied this steady state, students always have one very important misconception or myth in their mind. What is that concept? So for this, we will focus on the fine tuning of critical concentration. What exactly is this fine tuning of critical concentration? Critical concentration Cc refers to the concentration of ATP bound monomeric subunit of actin, that is G actins, above which polymerization will occur. So I will have a threshold beyond which polymerization occurs and below which the depolymerization would occur. So one of the common myths that most students after reading textbooks have, they always feel based on the treadmilling experiment that on positive end only addition occurs, that is polymerization, and on the negative end the depolymerization or removal occurs. However, the reality is that on positive as well as on the negative end, both polymerization and depolymerization can occur, and it is the fine tuning of the critical concentration which creates a condition where positive end grows and the negative end removes, or the removal process happens. Let's understand mathematically. We understand this mathematically. Okay. So we have the plus end and a minus end. The plus end is also called as the barbed end, and the minus end is also called as the pointed end. So dear students, let's clear this myth mathematically. Otherwise, our textbooks always tell us that the plus ends keeps growing and the minus ends keep shrinking. Let's calculate. First, let's focus on the plus end. And the plus end, we have the monomers where the polymerization takes place. It's 12 micromolar per second, that is the rate of polymerization, whereas the rate of depolymerization is 1.4 micromolar per second. So if you see the rate of in the plus end, the rate of polymerization is more than the rate of depolymerization. Similarly, let's focus on the minus end. So if the critical concentration comes to 0.12 micromolar, now let's see the minus end. In the minus end, the rate of polymerization is 1.3 micromolar per second, and the rate of depolymerization is 0.8 micromolar per second. So in the minus end, the rate of polymerization is again more than that of the depolymerization. The critical concentration happens to be 0.60 micromolar. So one thing is very clear, that both polymerization and depolymerization takes place from both ends, that is barbed as well as the pointed, or the plus end and the minus end, but then it is the critical concentration which determines. In precise terms, let's consider externally when we are adding salt, it is 0.3 micromolar. So now let's compare the plus and the minus end with respect to this 0.3 micromolar. If you compare the plus end is 0.12. So how much is 0.12 in terms of 0.3? 0.12 is less than 0.3. So therefore, at the plus end, there would be, so critical concentration refers to the concentration of ATP bound G actin, globular actin, above which the polymerization will occur and below which depolymerization. So here you see 0.3. So 0.12 and compare 0.3. So definitely the critical concentration is less than that of 0.3, and here 0.6, it is more than 0.3. Therefore, at the plus end, the polymerization occurs, and the minus end, the depolymerization occurs. To recap the major points once again, always you compare the critical concentration with the external concentration. Once you do the calculations based on the external concentration, let's see with respect to plus end as well as the minus end. With respect to plus end, the critical concentration is 0.12. So 0.12 is less than 0.3, the external concentration. Therefore, the plus end, there will be polymerization or there will be growth. minus end, the critical concentration is 0.60. This 0.60 at the minus end or the pointed end is more than 0.3. In other words, the external concentration in the minus end is less than that of the critical concentration. Therefore, the minus end will shrink or depolymerization would occur. Thus, if you consider it, the polymerization and depolymerization is possible on both ends, but it is the critical concentration and the interplay of the external salt concentrations which will depend that above the critical concentration the polymerization will occur and below it will be the depolymerization that occurs. In this segment, I shall brief you about the roles of various actin binding proteins. To simplify the task, I have taken this piece of information from a very prestigious journal, the Journal of Cancer and Metastasis Review. This review gives a comprehensive account of various actin binding proteins. At the very outset, we have actin nucleators. Under the category of actin nucleators, we have ARP23, VASP, NVASP and formins, of which in my lecture I will be talking about ARP23 and formins. Category 2 is G-actin polymerization proteins and F-actin depolymerization regulators. Amongst that, we have profilin, which features in my talk, thymosin beta 4 and cofilin. This helps in polymerization and depolymerization. The third category of protein is capping and severing protein, under which we have CAPZ and gelsolin. The fourth category is bundling and cross-linking proteins, for which we have FASN, filament and alpha-actinin. Anchoring actin filaments to the plasma membrane, this we will discuss when we discuss RBC membrane or erythrocyte membrane. We have talin, kindlin and ERM proteins. Of course, the major motor of actin, myosin, is the sixth category of the supporting binding proteins, and in seventh position we have F-actin stabilizing and regulatory proteins, the tropomyosins. Those are the stabilizing and regulatory proteins. First, we go to initiation and growth and actin filaments, under which we have formin. So, what formin does? Formin initiates and stimulates the elongation of actin filaments. You have a formin dimer that will lead to initial actin monomer. It will lead to considerable growth of the actin monomers till the G-actin forms a filamentous actin. But of course, for the conversion of ADP bound actin monomers to the ATP bound actin monomers, we have the role of profilin, that of course will come in the subsequent part. Then you have initiation and growth of actin filaments. Under that we have ARP23. This is the minus end or the pointed end. This is the plus or the barbed end. What ARP23 does, ARP23 leads to branching or branch formation. So, the branch formation is possible because of ARP23. The ARP23 complex binds to the actin filaments near the plus ends. You can always see this is the plus end, and even the branching is very much near the plus end. So, it branches near the plus ends and initiates the formation of branches. That was formin. Now, we have other proteins such as fimrin. If you see the diagram, these are parallel arrays. These are parallel arrays, and these fimbrian proteins, they are closely spaced actin filaments aligned parallelly, supports projection of plasma membranes such as microvilli. All filaments have same polarity, and the plus ends or the barbed ends are adjacent to the plasma membrane. So, these are the actin filaments shown in yellow and the fimbrian shown in orange, where fimbrian has a calcium binding domain. Next protein is alpha actinin. Alpha actinin binds to actin as a dimer. Each subunit is 112 kilodalton protein containing a single actin binding site. The filaments cross-linked by alpha actinin are consequently separated by greater distance than those separated by cross-linking of fimbrian. I will go to the fimbrian diagram once again. If you see the fimbrian, the distance of fimbrian was much less. Here in actin, the distance between the parallel arrays is much more. So, the alpha actinin filaments they are cross-linked by alpha actinin, are separated by greater distance. I showed what the greater distance implies, and when you compare the distance with fimbrian, it's much greater as compared to fimbrian. Why it is greater? Because alpha actinin, the length is 40 nanometer instead of 14 nanometer that for fimbrian. This mathematical figures, the 14 and 40, this itself suggests that the distance for alpha actinin is between the parallel arrays is much more than that of the fimbrian. Our next protein is profilin. As we discussed earlier when we were talking about formin, profilin binds actin monomers and stimulates the exchange of bound ADP for ATP, and this exchange promotes actin polymerization by increasing the local concentration of ATP actin. So, once the local concentration of ATP actin is more, the entire polymerization steps will be favored. Students, again, let's recollect the nucleation step. In nucleation step, we stated that ATP bound actin is more favorable for polymerization as compared to the depolymerization by ADP bound actin. So, that comes the role of profilin, and after profilin you have the proteins which is responsible for severing or breaking of this filament. Under that category we have cofilin. Cofilin is filament severing protein. As you see, filament severing, as the name suggests, the green is the minus end or the pointed end, the blue is the plus or the barbed end. Because of cofilin, the filament, the filamentous actin breaks into two parts, the minus end and the plus end is retained, and whichever end it breaks, that will form the respective opposite end. If it breaks, the minus end is there kept, you have the plus end, and likewise you have the plus end, and here you have the minus end. In very simple words, other actin binding proteins remodel or remodify or stabilize the existing filaments. Best example is actin binding protein cofilin, which severs the actin filament or which breaks the actin filament. Last but not the least, we also have stabilization of actin filaments. Under stabilization of actin filaments, we have plus end capping protein shown in blue, the minus end capping protein shown in green, and the actin filaments can be stabilized by capping proteins that bind to the plus or minus ends by filament stabilizing protein. One of the filament stabilizing protein is tropomyosin, and this tropomyosin can bind along the entire length of the filament by capping it on the ends. Let's understand the proteins associated with actin again through the animation from Cooper. In the cell, there is a formin. The two types of proteins are there. One is called as formin, which determine where the filaments are formed. Formin binds to the monomers and facilitate the nucleation process, in which three monomers are joined to form a trimer. After nucleation, polymerization spreads quickly. It is thought that these formins nucleate long unbranched actin filaments, such as those in muscle cells, thin filaments. At the leading edge of the moving cells, actin filaments treadmill and branch extensively. The branches are nucleated by arc-2,3 complex, which binds near the barbed ends or the plus ends of the microfilaments and forms a new branch. arc-2,3 helps promote the remodeling of the actin cytoskeleton that is required for cell movements and changes in cell shape. Having studied the entire dynamics and mechanism of polymerization, it's time for us to understand some of the toxins and their pharmacological mechanisms, how these toxins disrupt the actin monomer polymer equilibrium. We have two examples under actin polymerization and another two examples from actin depolymerization. Under the category of actin polymerization, we have jesplakinolide, which enhances the polymerization by inducing nucleation, as well as phalloidin, which stabilizes and compete for the same binding site. So, phalloidin and jesplakinolide. Then actin depolymerization, we have cytoskeleton D, which binds to F-actin at the barbed end, and lacturocolin, which binds to G-actin monomers. Both these molecules will stop the depolymerization process. Let's understand the organization of actin filaments. Basically, the actin filaments are organized into two basic networks. One, of course, is a bundle, another is the orthogonal filaments. The actin bundles, as you can see in the figure, you have several parallel arrays joined by small rigid proteins, and these small rigid proteins will align the filaments closely with one another. These are the small rigid proteins, and these small rigid proteins are aligned freely closely with one another. On the other hand, we also have, in contrast, a large flexible proteins, that is your network. Large flexible proteins, however, despite being large flexible proteins, they cross-link the orthogonal filaments, that's the actin bundles and the networks. In research, erythrocyte membrane is a very important tool. Erythrocyte membranes are studied by electron microscopy. It's extensively used as a tool. We have hypersensitivity reactions such as hypersensitivity reaction 2, which is directed against erythrocytes. So, erythrocyte is a very important tool in research. is an excellent tool, and some of the principal advantages of using erythrocyte membrane as a tool are: the erythrocyte membrane doesn't have nucleus or internal organelles. The plasma membrane and associated proteins can therefore be isolated easily in case of erythrocytes, and this isolation is possible even without contamination by various internal membranes that are abundant in the cell types. Another important point is, except for African camel llamas, the human erythrocytes lack nucleus. The nucleus, which develops even during the developmental process, the maturation process of the RBC, the nucleus is extruded. So, human erythrocytes lack other cytoskeletal components such as microtubules and intermediate filaments. So, since we can rule out microtubules and intermediate filaments, all that the human erythrocyte contains is actin, and therefore erythrocyte membrane is a very important tool for study of actin filaments. Let's look at the diagram. The association of erythrocyte cortical cytoskeleton with the plasma membrane, how the erythrocyte cytoskeleton attaches with the plasma membrane. We have series of proteins that will help in attachment. So, some of the associated erythrocyte cortical cytoskeleton with plasma membrane, some of the proteins. The major link between the spectrin and actin network and the plasma membrane is provided by the protein ankyrin. So, this is ankyrin. It provides maximal network between the spectrin actin network is ankyrin. Again, ankyrin binds to both the spectrin. This is your spectrin. So, ankyrin binds to both spectrin as well as ankyrin binds to both spectrin and to the cytoplasmic domain of an abundant transmembrane protein called as the band 3. So, there you have band 3. So, spectrin actin network is there. You have ankyrin. Ankyrin binds to both spectrin as well as band 3. Band 3 is the cytoplasmic domain of an band 3 is a transmembrane protein on the cytoplasmic domain, that's band 3. Then you have an additional link between the spectrin actin network and the plasma membrane, which is provided by ankyrin. This is spectrin and this is actin. So, spectrin and actin, the spectrin and actin together it provides, the plasma membrane is provided by protein 4.1. Protein 4.1, this is the protein 4.1. They are going to provide additional link to the spectrin actin network. Next we have protein 4.1 that binds to spectrin actin junction as well as cytoplasmic domain of glycophorin, which is another abundant transmembrane protein. Glycophorin is another important transmembrane protein. Okay. There you have glycophorin, which is another abundant transmembrane protein. Let's understand the structure of spectrin protein in detail. The major protein that provides the structural basis for cortical cytoskeleton in erythrocyte is the actin-binding protein spectrin. Spectrin consists of two distinct polypeptide chain, the alpha chain and the beta chain. both 2 alpha chains and 2 beta chains with respective molecular weight of 240 and 220 kilodalton respectively. The ends of the spectrin tetramers then associated with short actin filaments. This results in spectrin actin network, and this spectrin actin network forms the cortical cytoskeleton of the RBCs. What is the molecular motor? Molecular motor is a protein that converts chemical energy in the form of ATP to mechanical energy, thus generating force and movement. Our muscle, especially the muscle protein, the microfilament protein myosin, is also called as a motor protein. The best example is actin myosin interaction involved in muscle contraction. There are also examples of non-muscle cells involved in cell division, intracellular transport of vesicles. Motor proteins related to actin filament is just one example, that of myosin. First, what we will do, we will look at the structure of a muscle cell. If you take a cross section of a muscle, you have bundle of muscle fibers. The bundle of muscle fibers, we have several bundle of muscle fibers. From amongst those bundle of muscle fibers, you can zoom in or narrow down to one muscle fiber, that is individual muscle cell. Individual muscle cell will have nucleus, a plasma membrane, and within that you have one such myofibril, and each myofibril, when you take a cross section, you get what is called as A band and a I band. A stands for the dark band, that is anisotropic, and I stands for isotropic, and these indentations, right, these indentations are the ones between two, they would form one single sarcomere, that is between the two indentations or Z disc, and you have the dark band which is the A band. We zoom in further. So what is the arrangement of a sarcomere? Typical sarcomere, we have the arrangement, we have the I band and the A band. I, the light band, and A band is the dark band. So we have the A bands and the I bands. I bands of course are the light bands and A bands are the dark bands. Okay. So we have alternative dark bands called as the A bands and I bands, which are the light bands. Accordingly, if we see the structure of a sarcomere, we have centrally located A band. Once we see the diagram, we understand that anisotropic A are the dark bands, isotropic I are the light bands. Accordingly, we have towards the end a Z disc attached with a spring like protein, the titin that we see later, nebulins and titins. Thin filament shown in yellow is actin. Thick filament shown in red with several projectile head, they are the myosin. Again, we have the I band, the zone of overlap, H zone, and together the zone of overlap is forms the A band. So A band is also called as the dark band or anisotropic band. when the muscle contracts, this entire line would realign or disappear. Let's see how. So we have two sarcomeres. We have three situations: a relaxed muscle, a partially contracted muscle, and a maximally contracted muscle, of which the relaxed muscle, you can see this is a relaxed muscle. You have the A band, the H zone, the I band. In completely relaxed muscle, the myosin and actin are within their own loop, whereas in partially contracted muscle, the sliding has already started. The thin filaments slide over the thick filaments, whereas in maximally contracted muscle, the complete sliding has taken place, and you have the disappearance of a particular line just because of complete contraction. Focus on myosin. About 20 classes of myosin are there. They are designated on the basis of the sequence of amino acids on their ATP hydrolyzing motor domains. These myosins are present both in animal and plant cell, as well as in muscle as well as non-muscle cells. Myosins are large superfamily of motor proteins that move along actin filaments while hydrolyzing ATP. Let's focus on the structure of myosin 2. The structure of myosin 2 consists of two yellow colored essential light chain and two green colored regulatory light chains. So total there are 4 light chains, 2 essential light chains and 2 regulatory light chains. We also have a heavy chain. In fact, there are 2 heavy chains are coiled by alpha helical tails. Myosin 2 move from plus N to minus N, whereas myosin 6 move from minus N to plus N. Together, actin plus myosin is responsible for muscle contraction. What's the organization of actin and myosin filaments? As we know, the border forms the Z disc. You have the thin filament actin shown in yellow, the thick filament myosin with projectile heads which lattice across to actin, and actin in turn is intertwined with nebulin, and towards the Z disc we have the spring like protein, the titin. So these are some of the essential proteins of muscle contraction, and again we have a plus end and a minus end. We have a distinct plus end and minus end for the organization of actin and myosin filaments. Now, what is the association of tropomyosin and troponin with actin filaments? We have calcium. Generally, when you talk about the tropomyosin, the troponin complex consists of troponin I, troponin C and troponin T. Now I shall brief you about the structure of actin, myosin and some of the proteins which are associated with thin filaments. At the very outset, let's understand the association of tropomyosin with that of troponins and actin filaments. Let's understand the color coding first. The actin filaments are shown in yellow. This brown rope like structure which you can find is intertwined, or just like a creeper, it turns around the actin. This is tropomyosin. As I said, the yellow colored is actin. Not shown in the figure, If you see this particular figure, on each actin where I have highlighted just now, on each actin you can find a central nucleus or rounded structure. That's exactly the structure or which is the binding site for myosin. However, what happens, as you see, when there is absence of nerve impulse, this binding site of actin remains blocked. Now, what blocks this active site? This active site is blocked by tropomyosin, the brown rope-like structure that you see. The tropomyosin will block the active site, preventing myosin from binding. So, myosin binding site is blocked because of the covering of this particular site. Now, when there is a calcium, nerve impulse takes place, suppose there is calcium, what calcium does, calcium will lead to a change in conformational structure. The tropomyosin, which was otherwise blocking the myosin heads from lodging with the actin, that block is removed. Once the block is removed, the troponin complex together with myosin binds to the actin. Troponin complex, student, is made up of three molecules: troponin I, troponin C and troponin T. So, tropomyosin binds lengthwise along actin filaments, and in striated muscle it is associated with complex of three troponins: troponin I, troponin C and troponin T. What's the association of tropomyosin and troponin with actin filaments? In the absence of calcium, just now what I explained, the tropomyosin troponin complex, this is tropomyosin, this is troponin complex, this entire complex blocks binding of myosin to actin. That is, the rounded structures are not exposed. But once calcium binds, the troponin C shifts the complex, and after shifting the complex it will relieve the inhibition, and post relief of inhibition it will allow contraction to proceed. What are the proteins that are contained in thin filaments? Some of the proteins contained in thin filaments are actin, nebulin, tropomodulin, tropomyosin and troponin. First we will study tropomyosin. As we know, tropomyosin keeps the actin heads dislodged so that myosin cannot join. Tropomyosin are the protein molecule which bind with the actin filament grooves all along the length. Coming to troponin, troponin also is a protein molecule which binds along the length of the actin filaments. These troponins are the combination of three polypeptides: troponin T, troponin C and troponin I. Troponin T binds to tropomyosin, troponin I binds to actin and troponin, troponin C is an analog of calmodulin, therefore it binds to 4 calcium molecules. Again, when you talk about troponin I, it binds to actin and troponin, and after binding with actin and troponin T, It inhibits the interaction between F-actin and myosin. Now we study nebulin. Nebulin is a structural protein. First of all, this is nebulin, the purple colored intertwining structure. Nebulin is a structural protein which bind along the length of the actin and helps in regulating the length of the filament. It stretches the actin filament from the ends, that is your Z bands, to the minus end. Minus end is of course towards the M line. Tropomodulin. Tropomodulin is a protein which stabilizes the actin filament in the sarcomere by capping minus end of the actin. What are the proteins related to thick filament? Myomycin is a protein that binds to the thick myosin filament at the H zone to form M line. Titin is a molecular spring. It keeps it midway of the Z disc. We also have contractile assemblies in non-muscle cells, not just muscle cell which I talked. Try to understand, even in non-muscle cells, you have bipolar filaments of myosin 2 which produce contraction by sliding actin filaments in opposite direction. This also leads to the contractile feature of a muscle, and especially a non-muscle cell. Finally, we have the regulation of myosin 2 by phosphorylation. In the very first step, calcium binds to calmodulin, which in turn binds to myosin light chain kinase, that is MLCK. The active calmodulin MLCK complex then phosphorylates the myosin 2 regulatory light chain. This is how the regulatory light chain is phosphorylated. You have phosphate addition, that is ATP is converted to ADP. Thus, the active calmodulin MLCK complex, that is myosin light chain complex, phosphorylates the myosin 2 regulatory light chain. Once the regulatory light chain is phosphorylated, it will convert myosin from an inactive to an active form. So, it will convert inactive myosin into an active myosin. The machinery of skeletal muscle includes thin and thick filaments. The machinery of skeletal muscle includes a thin and thick filaments. At the core of the thin filament is a actin filament. Based on the orientation in which the subunits called globular actin molecule attached to each other, the site where a protein, a myosin thick head, can bind. However, in the absence of a nerve impulse to stimulate the muscle fiber, a long third protein called as tropomyosin lies blocking the myosin binding site. 3 polypeptides in the complex called troponin associate with actin and tropomyosin. Troponin, tropomyosin and actin constitute the thin filament. Troponin can move tropomyosin and unblock the myosin binding sites. A cross section shows how troponin and tropomyosin associate around the actin and block the binding of myosin head. A nerve impulse triggers skeletal muscle cells to release calcium ions into the cytoplasm, an event that initiates muscle contraction. Calcium ions bind to troponin C polypeptides, which shift the position of the troponin tropomyosin on the actin filament, allowing myosin head and begin muscle contraction. After some time, calcium levels become low again and troponin and tropomyosin shift back, covering the myosin binding site, causing muscles to relax. This concludes, along with animation, the entire process of myosin and actin mediated muscle contraction. To summarize this segment, we have thin filament and thick filament, as well as myosin. Myosin is the major motor protein of microfilaments, that is actin. Since we have already completed microfilaments, especially actin, with all paths being complete, it's time for us to focus on some of the MCQs which have been asked in previous entrance examination. Here goes the first one. The phenomenon that illustrates the dynamic behavior of actin filaments and is critical to regulating the structure and function of actin filaments is known as, of course, we have four options: assembly, dynamic instability, treadmilling, and disassembly. The dynamic behavior refers to the constant where the rate of polymerization equals to the rate of depolymerization. So based on that, the correct option shall be option C, that is treadmilling. Actin filaments are stabilized by four options: cofilin, gelsolin, thymosin, and tropomyosin. Of course, just now we saw that in muscle contraction you have the binding sites for thick filaments, that is myosin, and those myosin sites are blocked, and they are stabilized unless of course the calcium comes. So the correct answer of course is tropomyosin, that is option D. The next question: branching of actin filaments can be initiated by. This question is from the section of the various proteins associated with actin, and the correct answer which initiates branching is option A, ARP23. Latrenculin blocks the dash. This is from CSIR Net June 2013. Option A, plus end of microfilaments. Option B, minus end of microfilaments. Option C, actin monomers. And option D, actin filaments. This is a direct question. Just to make you remember what we studied, the toxins that disrupt the actin monomer polymer equilibrium, we had two under polymerization and two under depolymerization. Here actin depolymerization, the two toxins are cytoskeleton D and latrenculin A. So latrenculin A disrupts the actin depolymerization. Option C, that's actin monomers. That concludes the entire section of microfilaments, that is actin.